Professor John Robertson, thank you very much for giving us some time at the National Cancer Research Institute. You've just given a really interesting talk in a first-class session on breast cancer. What was it about?
Well, what I was talking about was what I think might be the future in terms of early diagnosis of breast cancer, but not only breast cancer, but potentially all solid tumours, which is based on the fact that people make immune responses to their cancer, B-cell immune responses, and we believe now that they make that at a very early stage. When people present symptomatically, usually the cells, probably at 35 cell divisions out of the 40 before you die, and even with screen-detected cancers, it's about 30. So there's a huge area between 0 and 30 whereby we might be able to find a cancer, or identify it's there and intervene. But from previous work I've done a number of years ago looking at cancer antigen markers, you can't detect those until it's too late. Usually by the time you see an elevation of the marker, you've got metastatic disease. And in fact, we talk about the marker being abnormal, we say it's above the limit of normal, which means that you get this antigen produced from normal cells too. Take CA15-3, for example. The implication of that is that the mouse monoclonal in the kit can't tell the difference between the normal cell-derived antigen and the cancer cell-derived antigen. And the implication for many people from that has been that they're the same. And in fact, they're not the same. You get abnormal glycosylation, you get mutations, etc. And the body is exquisitely sensitive to what is not self and makes an immune response to that. And so you should be looking for an amplification signal to the immune system, which you don't see if you're simply looking for the antigen itself.
So, what's your trick?
Well, the trick is to be able to do this reproducibly, you know, and in the past there's a lot of data in the literature about the science of this, that there are antibodies, and I think that's not in any doubt. And there's also data in the literature that you can detect these antibodies months, if not years before. For example, one of the areas we've been working on recently is lung cancer, and there's a publication in 2006 suggesting that you can find these antibodies up to 5 years before screening CT can identify those patients. But I think the important thing for any clinical test is its reproducibility. And we spent a lot of time working on the technical aspects of that.
How do you do it?
Well, it's good science and also a lot of technical work. You know, for example, how do you calibrate a polyclonal response in cancer? That's not an easy thing to do. It's easy to do to calibrate a monoclonal with a monoclonal antibody to calibrate an antigen, but it's not easy to do calibrate a polyclonal response. And we're about to publish that data, so it's it will come out soon.
How would you validate this then? You've got to get patients who've got BRCA1, 2 mutations and follow them.
Well,
heavy smokers,
yeah.
colon cancer, pre-malignant syndromes. How are you going to do it?
Well, I think the first thing to do is to show that you can diagnose early stage disease, which antigen markers have not been able to do. And I think that there's some data in the literature, and we've now produced data with over about 600 cancers where we're going to be able to show that. The next thing to do is to show that you can find it in early stage, you know, pre-diagnostic. And so I think there's a lot of samples that are available without having to do prospective trials necessarily.
Is it too early for the patient to read about this in the national newspapers?
No, I don't think it is too early. I think that the test could be useful alongside other imaging technologies. And in fact, in the US, I think that's probably the place where it may first become available, where CT screening is already used by some people, but not yet, I think, established. Then a blood test to help CT screening, as an aid to CT screening, may be a useful adjunct.
Well, in the European Institute of Oncology in Milan, we're just expanding our CT spiral group from 5,000 to 15,000, so we should maybe talk about that.
Yep.
John, thanks very much indeed.
It's a pleasure.
Good.